name is Dion Rossiter. I am the Executive Director of Science at Cal. Rosalind Sarvey is our Public Engagement Specialist. I'm going to actually hand things over to her to take over, but we have an amazing program for you today. So Rosalind, take it away.
Thanks so much, Dee. Like Dee mentioned, my name is Rosalind Sarvey. I'm our Public Engagement Specialist. Just want to thank you all for being here tonight. We've got a really exciting program with two of our researchers at UC Berkeley, Nina Maryn and Taormina Lepore. So let's go ahead and get started. Just want to start off the night with a land acknowledgement to recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So thank you for allowing me to give that land acknowledgement. So for those of you who are new or you've been with us for a long time, Science at Cal brings the wonder and excitement of UC Berkeley STEM research to the community. All of our programs and events are free and geared toward public audiences like yourself. So thank you so much for being here. We celebrate science through our science cafes, our lectures, festivals, Cal Day, and so much more. We're usually out in the communities and on campus. Of course, we can't be here in person with you at the moment, but we thank you for joining us online. We're also planning to be in libraries and farmers markets hopefully soon. So make sure you check out scienceatcal.berkeley.edu for more of our upcoming events. For any of you who have donated for this program and our other programs, thank you so much for supporting all of our programs and events, especially our advisory council and members of the VCRO. We thank you, and we thank the Lawrence Hall Science as well. Save the dates for our upcoming events. We have our Midday Science Cafe in partnership with Berkeley Lab. We've got 3 events this summer. One's actually next week on synthetic biology. So be sure to sign up for our newsletter so you can check out all of our upcoming events, as well as our lecture series. We have finally our first event on COVID-19 is actually this month, and that'll be with Dr. Karen Nelson. So make sure you save the date for that as well. And then our Grounds for Science event, of course, we have 2 other amazing programs coming up this summer, so be sure to save the dates for those as well. So for any of you who have made our cocktails for this evening, these were inspired by our presentations for the night. We have a citrus Campari spritz inspired by Nina's presentation on kind of a botanical theme, and then also our fossil affogato elixir, which is inspired by our other presentation this evening, which has a milk product in it. So let us know if you're drinking any of those tonight. To give you a little bit of a rundown of how this evening will go, we're actually going to start off the night with a round of trivia, which is focused on milk. So we'll go ahead and get started on that in just a minute here. And then we'll actually have our first presentation with Taormina, and she'll be giving her talk. And then we'll do probably a little bit longer of a Q&A than we normally do, because Taormina is actually at the airport right now and she may have to go catch her flight. So if you have any questions for her, be sure to type them into the chat as she's giving her presentation, or you can actually go ahead and kind of interrupt her during her presentation. She's let us know that she's okay with that. Then we'll go ahead and go into our second round of trivia. And then Nina Maryn will go ahead and give her presentation on why don't plants get sunburns. We'll do another Q&A with Nina after that. And then there will be some more time at the very end for a longer Q&A if you'd like to stick around for that.
All that to say, everyone, as we're going through these trivia questions and also the presentations, make sure you add your questions into the chat. As many questions as you can. We'll try to get to all of them. So let's get us started, Rosalind, on trivia question number 1 for Taormina.
Thanks so much, Dee. One thing I forgot to mention is that this is actually being recorded. So for anyone who wants to catch this presentation later, it will be recorded and available to you afterward.
True, true. All right, so question number 1. What do you guys think? Which group of vertebrates also produce a liquid nutrition product, which it regulates to its babies? Oh, regurgitates. Oh, wow, I had that way wrong. Regurgitates. Which group of vertebrates also produces a liquid nutrition product, which it regurgitates to its babies? We're going to try to get up to 80% of people participating. So.
Wow, it looks like we kind of almost have a consensus here.
Yeah. Reptiles, birds, amphibians, fish. What do y'all think? Let's get a few more people voting. Come on, guys. Let's see how much you know. You're going to go into this presentation. All right. I think we're good. If you want to end that poll, Rosalind, let's see what people picked. Ooh, birds. So let's see. Taormina, you there?
Hey, yeah, Dee, I'm here.
Do you want to talk about this? Seems like people knew the answer.
Yeah, so I'm super impressed. We got a good chunk of folks responding birds. And that is the answer that I was looking for. So birds actually produce something called crop milk, CROP. And crop milk is something that they regurgitate. It's not quite like milk from mammals, but it is a nutritious product that's produced for some bird babies. Pigeons do this, and flamingos as well, and some pigeons. Some penguins. So yeah, great job.
Good job, everyone. All right, let's go ahead to our second question. All right, so lactose, also known as a milk sugar, likely persisted in certain adult populations as human societies developed.
Is this missing the word tolerance?
Yeah, I'm wondering if this is missing a word. This doesn't seem to make sense.
Yeah, it should be lactose tolerance. Yeah.
Tolerance. Nice. Okay. Lactose milk sugar tolerance likely persisted in certain adult populations as human societies developed.
Looks like we've got about half of our participants. Keep the votes coming.
Yeah, we really are not seeing a consensus here.
Tricky one.
Yes.
All right, we're just
right.
out there.
Oh, let's see what people said. Pastoralism. And is that the answer, Rosalind? Yes.
It is.
So, Taormina, what do you have to say about this?
Yeah, so really this should be, and that's my bad, you all, lactose tolerance or digestion, perhaps, likely persisted in these adult populations. As human societies develop pastoralism, which is the caring and raising of cows. So agriculture is a really good guess here too. And you could maybe put pastoralism under the category of agriculture, but pastoralism's the better answer here. So great job, everyone.
Excellent. Thank you. And our third question, when did the first mammals most likely evolve based on fossils? 240 million years ago, 66 million years ago, 50 million years ago, or 300 million years ago?
I think this one might be trickier for folks. The answers aren't coming in as quickly.
Yeah, that's okay.
And they're also there's some debates amongst our audience. All right, go ahead and end that poll, Rosalind. Let's see what people say. Are you sharing results, by the way? Oh, you are sharing. Okay, so we have 66 million years ago at the end of the age of the dinosaur. And what is the correct answer? Let's find out. 240. Taormina?
Yes.
Mhm.
Yeah, so this is a real tricky one. And the question is phrased in a way that might make people kind of second guess themselves, especially with the age of quote unquote mammals being maybe tricky, or the age of, at the end of the age of dinosaurs. But based on fossils that we have of the earliest mammals and their ancestors, we think that a good consensus is around 240 million years ago, at least closer to the beginning of the age of dinosaurs than closer to the end. We get our first mammal-like things and our first true mammals closer to the Jurassic period. Yeah, just after 240 million. So hope that helps us think about our ancient past a little bit more.
Yes, and thank you everyone for participating. And actually, this is a really great lead in to Taormina's presentation. So we're going to go ahead and get started and give her the floor. And I'm just going to do a quick introduction. Taormina Lepore is a second-year PhD candidate in integrative biology at Cal. She focusing on vertebrate paleontology and evolutionary biology. She grew up outside of Boston in a big Sicilian American family with strong cultural roots. Taormina became interested in science as a little kid, reading lots of dinosaur books and collecting spotted salamanders in the marshy areas of Massachusetts. She identifies as a disabled, queer scientist, and is coming back to a graduate program after a decade as a high school science teacher. And I know we have some high school students on this video program, so it's so happy this is appropriate that Taormina is our first presenter and you're all here. She's fascinated by the evolution of parental care and the special nutrition information recorded in fossil teeth. Her work as a science education researcher also focuses on the use of STEM education in the classroom and outdoors as a vehicle for increasing inclusion in science. She is really excited to join Grounds for Science through Science at Cal, and we are very excited to have her. Thank you so much for being here, Taormina. And remember, everyone, ask your questions in the chat box, or we'll give you a minute to you don't even have to answer your questions in the chat box. We didn't say this either. At the end of our program, if you want to unmute yourself, you can ask her your questions yourself. So go ahead, Taormina, and take it away.
Thank you so much, Dee. Thanks, Rosalind and Nina. And thank you all for being here. I want to apologize because I am at the airport gate, so there's a lot of background noise. Hopefully my connection sticks around. But thank you all so much. Again, I'm Taormina. I'm a PhD candidate in Leslia Lusko's lab here at Cal. And I'm about to get on a plane to go back to the San Francisco area. So my topic today is got milk. And I don't know if you remember those old commercials about milk that used to say got milk, but maybe those commercials, if you ever saw them, might have made you think, well, how can we tell milk first came around? And so the topic today will be, how can we tell when our mammalian ancestors first produced milk? And it's kind of a mystery. So where are we headed today? First, I'd like to just briefly talk about, what is milk? Why does milk matter? Can we figure out when milk evolved? What's the evidence that we might be able to find about milk evolution? Is there a connection between milk and teeth? Sometimes growing up, we may have heard that milk is good for our teeth and bones, but is there a connection there? And where do we go next with this work? So what is milk? Milk is a nutritious product. It's liquid. It has everything a growing body needs, and growing baby humans, as well as growing mammals, all mammals, feed on milk. They drink milk during the early years of their lives. And so all mammals do this, including humans. But overall, we can say that milk produced by mammals is energy. So it provides that energy for the growing baby. Milk is nutrients, so it's the food that the growing baby or baby animal eats or takes in. Milk is a water source, and so you'll see in a couple slides how much water is in milk. And milk is a great immune system booster. So without milk, we wouldn't have an immune system really early on as babies, at least one that isn't as strong. And so it can really be a lifesaver for young babies and young mammals. So what's the stuff that makes up milk? Well, if we take a big pie chart, milk is about 80% water. And this pie chart is actually cow milk, so milk from a cow. Human milk is not too far off from this. So 88% water, that really is a thirst quencher. And the remainder of the pie is about 3% proteins, especially one particular kind of protein only found in milk called casein. In addition to casein, some other proteins in milk are immunoglobulins, which are specialized immune system boosters that really help the immune system grow, like I mentioned a moment ago. The other 8% or so is specialized fat, especially milk fat, special sugars like lactose sugar, unique to milk, and about 1% of different vitamins and minerals. So calcium, barium, vitamin D, and some other good minerals that really help the body grow. But why does milk matter? I mean, maybe some of us don't drink milk, or maybe we only drank it when we were babies. Why does it matter? Well, all human beings drink milk or some kind, they drink milk or some kind of milk-like product when they're babies. Milk is also produced by cows as an industrial product around the world. So we milk cows and drink cow milk, and also sheep and goat milk and some other milks. It's connected to the development of humans and cows together in this concept called pastoralism. Milk also ties us back to our mammal ancestors. And these cute little guys are alive today. They're living animals called echidnas. Echidnas are marsupials, or excuse me, they're monotremes like a duck-billed platypus. And they lay eggs just like a duck-billed platypus. They live in Australia. And the echidna doesn't have nipples. It actually oozes milk just straight off of its body. So that's a very interesting factoid about how milk first developed. It wasn't coming out of nipples at first. And milk throughout the process of a baby feeding on milk, throughout the time that it's feeding on mom's milk, milk will have different appearances and different material inside of it that will help boost the immune system or provide more water and so on. So you can see in these two pictures here, this is human milk, and one on the left-hand side is a little bit later on in milk feeding, and the thicker looking one is a little bit earlier on, right when babies first start to feed. So thinking about why milk is important and all of this cool stuff about milk, can we figure out when milk first evolved? Well, in order to get at this question, we have to think about the animals that have milk today, milk like we know it in mammals. There are two major groups that I'm going to mention here. The one on the left-hand side is a big group of organisms called sauropsids. Sauropsids living today include birds and reptiles. And in the fossil record, we also have some specialized fossil reptiles. And on the right-hand side, the other group that split away from the sauropsids are called the synapsids. And synapsids include all mammals living today and their fossil relatives, and some things that don't even look like mammals, but look a lot like reptiles, but they are in the synapsid group. Now, this split in the family tree between the birds and reptiles and the dinosaurs over there as well, and on the synapsid side, the mammals and their relatives, we think happened about 310 million years ago. So that gives us the oldest time possible for milk to evolve. But there's a problem. Milk doesn't fossilize. Now, we have some really cool fossils from places like China that have excellent preservation. And this fossil here is of an early mammal from the age of dinosaurs. And the black stuff all around the skeleton is actually fossilized fur. But even though we can get fossilized fur, we can't quite get fossilized nipples or breast tissue. So what are we going to do? Well, we could look at genes as well. And if we take the fossils and genes together, genes that make up milk and genes that make up yolk that make eggs, we could try to put some more barriers in place to say, okay, well, when could milk have evolved? Today, mammals don't lay eggs, so they don't have yolk genes. But there are special mammals that do. There are some exceptions, like our echidnas and platypuses that lay eggs. So if we look at when the split for these groups happened, and when yolk in the eggs started going away, and when the first mammals appeared, we can say, well, you know, we don't have yolk anymore. So it had to be after 210 million years ago when that genetic evidence appears. And we think that somewhere between 310 million, that split, but before 180 million, the first mammal fossils with fur, milk must have been around. Okay, so somewhere in that big span of time. So what else can we do? And I mentioned early on that there might be a connection with teeth. Well, teeth grow just like trees. They have lines in them, just like lines in the inside of a tree. And we might be able to look at the inside of teeth to figure out when milk feeding was occurring. We call this kind of field paleodentistry. And you can see this slice of a tooth here. If we were to look at the inside, we can look at the chemistry of the inside of the tooth and these special lines, just like tree rings, to try and count, okay, well, how many lines equal milk feeding? And if we look at modern humans today, if we take a baby tooth and slice it up and look at the inside, we can tell exactly when that baby started and stopped feeding on mom's milk. So I'm really curious to look at fossils in the fossil record and take a look at these teeth and say, can we see similar signatures in the chemistry and the lines of these teeth? There are other studies that look at these lines of teeth, and you can sort of see that they do look just like tree rings. This is a tusk from a mammal cousin that's been sliced, and you can see the highlight here has these lines of growth. Now, this study wasn't about milk, but it was about hibernation. So teeth can really tell us all kinds of cool things about the metabolism of ancient animals. So where do we go from here? In the last couple minutes, I just want to make sure I mention teeth can be really important because they stick around in the fossil record. They're very good miniature time capsules of an animal's metabolism of their life when they were alive. But if we just look at the outsides of the teeth, the baby and adult teeth, it might not be enough evidence. So going into the inside of those teeth, looking at those tree rings of growth and some of the chemistry might help us unlock when the earliest mammals were truly starting to feed on mom's milk. So we can narrow down that 310 million years to 180 million years time frame to something a little narrower, I hope. So my plans include looking at some teeth at the UCMP, UC Museum of Paleontology. And in order to do that, I basically take teeth and I put them in some plastic. You can see pouring the plastic here. Then I take a special saw and once the plastic is hardened with the tooth inside of it, I very carefully slice that plastic block with the tooth in it. And then I can make a little microscope slide and stick a very thin slice of tooth onto that microscope slide. And when it comes out, when it's all done, hopefully it looks like a nice slice of tooth, just like on a deli slicer, like slicing deli meat. And I can look at the inside of those teeth for some lines of growth. I'm also hoping to go and work at some other places like the Royal Ontario Museum and the University of Kent in England to try and get some more training. And so I wanted to mention that because it's really important to ask for help from others when you're getting started as a scientist. I have some image sources. And ultimately, yeah, I just want to thank you all so much for listening. Thank you to my lab members, my advisor, the entire team here for organizing science at Cal. Thanks so much.
Thanks so much, Taormina. That was wonderful. We did have a few questions coming in as you were presenting, and I think I actually saw Dale had a hand up. I don't know if Dale, you wanted to go ahead and ask your question. If you want to unmute yourself. Maybe not, but that's all right. If anyone has a question, you want to raise your hand, feel free to do that, and then you can unmute yourself. But we did have a couple of questions come in through the chat. One question that we got was, does baby formula contain immunoglobulins to mimic the immune boosting properties of milk? Is that something you can kind of talk about?
Oh, sorry, I was looking for the mute. That is a really good question. So, so basically, there, to my knowledge, the immunoglobulins that are in breast milk are kind of not the same that you could get in formula. But I really don't know. Like I haven't had kids myself. I don't know if there are any immunoglobulins at all in formula. I just know like that the breast milk immunoglobulins are super unique and really helpful to provide a boost of immunity during the early stages of a baby being born. So that's a great question. I'd have to look that up.
Thank you so much. We actually got kind of a follow-up question, and maybe you can backtrack a little bit. What is an immunoglobulin, and how does that boost the immune system?
Yeah, great question. So maybe you've heard of something called an antibody. And an antibody, we have lots of them all around the body that help our body determine signatures of disease and attack the disease. So immunoglobulins are one type of antibody, and it's really important for babies to have a big boost of them because fresh being born, their immune system doesn't have a lot of that innate immunity. And so having those antibodies from mom's milk is really a great survival mechanism.
Hey, Taormina, before you answer the next question, can you put your slides back up and go into the slide with the image sources? And leave that up as you answer the next question, because somebody wanted to see that slide. So sorry to interrupt, Rosalind. Go ahead with that next question.
No worries. Thanks, Dee, for watching the chat there.
Yeah, and also we will remind everyone that, you know, we could also provide some of these sources on our website when we go to send out. You guys will all get the video recording, and we can add these to the page if that's helpful for whoever would like these image sources as well.
Absolutely. We did get another question about pasteurization and if that destroys the immune boosting properties.
Yeah, that's an excellent question. So I'm still learning a lot about milk and how milk is processed. I really don't know if you were to pasteurize, say, brand new, just produced breast milk, human breast milk, if that would destroy the immunoglobulin properties. Yeah, when cow's milk is pasteurized, that process is meant to get rid of a lot of microbial stuff. So the short answer is I don't know. The longer answer is I think it depends on whether we're looking at human milk, cow milk, and if we could get some information on either of those, that would be very interesting. Just to follow up on a previous question, I just glanced at something here, and it looks like there are some baby milk formulas that are being sold infused with prebiotics, which might be an attempt to kind of replicate the immunoglobulins that would naturally be produced by mom. Yeah, great questions. If I can't get you an answer right now, feel free. We can also, you know, you're welcome to email me as well. I love these questions.
Yeah, these are great. Keep them coming. We had another question, Taormina. You know, you described part of your work right now is figuring out the direction that you want to go. What could happen if you don't find weaning signatures in fossil teeth?
Yeah, it's a great question. So right now I'm at the part of my work where I'm really trying to get something called pilot data. And pilot data is just the beginning data that we look at. And so if I can't get weaning signatures in teeth, number one, I might look at that and say, well, maybe that's, you know, this is a true absence of a weaning signature. Or I could take a look at the teeth that I've sliced and look at other things that are preserved metabolically. Like another thing I'm really interested in is a birth line. It's actually a line that we all have when we're born that is recorded in the first molar. So our first adult molar, so the 4 of them that we have, records this line of birth. So that's super interesting to me too. So I'm hoping I'll get something interesting out of these toothy slices.
Thanks so much for sharing. We had an interesting question in the chat that I wanted to share with you, and it's what makes something milk versus juice? For example, should we say almond milk or almond juice?
That's a great question. I sort of think in my snarky kind of way that the almond and hemp and coconut milk kind of terminology has sort of stolen milk as mammal milk. But then again, we could think of it in a big umbrella kind of way. Like maybe milk is just a milk product or a milk substitute, and it's okay to call it milk. If I were to take my like super specific like sciencey way of thinking about it, I would actually include milk as the stuff that those birds, the example with birds that they throw up, that might be a milk. What mammals produce as milk, and also something that some amphibians produce from basically where they lay eggs, like the egg opening. There's another type of fluid. I would call those things milk as nutritional products for babies before I would call almond juice or hemp juice. But I still buy it, and I don't scratch it out on the carton or anything. So.
Thanks so much for clarifying. I know, there's a lot of talk about that at the moment, especially. So I know you have to get running pretty soon here to catch your flight, but we did have one last question for you here. And someone mentioned, I've seen mothers who have received the COVID vaccine, giving their children the milk with hope to pass on that immunization. Does that work?
That's an excellent question. I don't have the answer to that question because there's so much that's changing right now with the research that's going on with COVID. But I have a lot of friends who have young children who have gotten the vaccine. And so just anecdotally, when I'm asked things like, does that work? I think it really is up to each individual person to know what's best for them and their child. But at the same time, the vaccine's super important. So like, what do you do? I would trust the advice of your doctor. I would trust your intuition and trust science. But I also don't have an answer because I don't know who has an answer to that kind of question. It's a great question, but I think that'll be something really interesting for us all to think about is how long does COVID immunity stick around in things like breast milk, if at all?
Absolutely. Well, thank you so much, Taormina. I know you have to go catch your flight, so I just want to thank you again so much for being here and for the fantastic presentation. We had a lot of great questions coming in, so thank you again so much. We'll let you know if there are other questions in the chat. We can definitely get those to Taormina, so feel free to let us know if anyone in the audience has other questions. So thanks again, Taormina.
Thank you all. I hope the background noise wasn't too bad. I appreciate it.
All right. And then with that, we're going to go ahead and jump into our second round of trivia. So Nina, if you are there, we are going to go ahead and get started with our plant trivia for the evening. So let me go ahead and bring that poll up here for you. All right, so our first question is, what wild plant is broccoli, cauliflower, kale, cabbage, bok choy, and Brussels sprouts all domesticated from? That's an interesting question. All right, we've got quite a few answers coming in. It looks like there's actually one that's kind of leading.
I know, and I had no idea what the answer to this was. You guys are smart. We got a smart audience.
All right, it's getting a little more mixed now, so
Yeah.
we'll see. This is an interesting question. It looks like we're almost getting everyone's answers in. Let's go ahead and end the poll here. All right, so it looks like most people went with mustard. Nina, is that correct?
It is correct. All of the cruciferous vegetables, which are those dark green vegetables like kale and bok choy and Brussels sprouts, they're all actually domesticated from the exact same wild plant, which is wild mustard.
Very cool. I had no idea. I knew those were all in the same family, but not related to mustard. Very, very cool.
Yeah, it's amazing. Very quickly in domestication, you are able to create all different kinds of plants that all look and taste very different from each other.
All right, let's go ahead and launch our second trivia question here. What percentage of soybean growth in the US is genetically modified? Is it 4%, 34%, 64%, or 94%?
We got one taking the lead.
Yeah. All right, looks like we got a lot of people bringing their answers in. I'm going to go ahead and close the poll here.
We have no votes for.
No votes for 4%. A lot of people said
I'm very impressed. Yeah, I'm very impressed. 94% is the answer. I would have expected most people to be a little more conservative and pick something more in the middle. But yeah, 94% of soybeans grown in the United States are genetically modified. They're predominantly modified for herbicide resistance so that they are not susceptible to the herbicides that are used to kill weeds that might interfere with growth. This actually allows fewer pesticides and fewer toxic pesticides to be used in farming. But the vast majority of the soy grown in the United States is just used for animal feed and it is not used in human feed, if you will.
Fascinating. Thanks so much for sharing more about that. It's so interesting. Let's go ahead and close up the trivia with our last question. And it's, what was the first GMO approved in the US used to create, I think I may have messed up this question here. Do you want to go ahead and remind me what
that was right. So the first GMO approved in the United States was used to create something. Okay. So what was that thing?
You know what it was? So, I tried to put a blank, like a line in the question, and then Zoom didn't put that in. So you're filling in the blank here, kind of.
Yeah.
All right, it looks like about half of everyone has responded, and we're actually getting a very split response here. It looks like almost 50-50. All right, maybe this one really stumped people. Let's see.
I love there's another one that got 0.
No one thought CRISPR?
Right,
looks like most people answer corn syrup.
So that's actually not correct, but it's a very good guess because a lot of corn in the United States is used to create high fructose corn syrup. But the answer is actually the first GMO in the United States was approved in 1982, but created in 1973. And it was a genetically modified bacteria called E. coli, and it was engineered to create human insulin, which is used as a drug to treat diabetes. And prior to that, people had to create insulin in a much more cumbersome way. And on the left, I actually have a factory assembly line where people were manufacturing insulin in a factory.
Very, very cool. All right. That ends our trivia for the evening. Thanks so much for sharing your answers there and learning more about plants. There's a lot of fascinating research going on out there. So this actually is a perfect way for me to go ahead and introduce Nina. So I'm going to go ahead and stop sharing my screen here and introduce our next speaker. So Nina Maryn is a National Science Foundation Graduate Research Fellow and PhD candidate in the Neogi Lab at UC Berkeley. She studied biology at NYU, where she researched dosage compensation in worms. And Nina wanted me to mention that these are very, very small worms, not earthworms, and spore formation in bacteria. She now studies photosynthesis and how it can be improved using the biodiversity of the natural world in order to someday help grow more food with fewer resources. Her major concerns are climate change, bad urban planning, and inconsistent bus schedules. In her spare time, she enjoys writing short stories. Hopefully soon there will be one coming out. Performing live stand-up comedy and game mastering role-playing games for her friends. She's interested in doing more work on engaging with the public on issues of scientific ethics and expanding access to scientific literacy during her PhD and beyond. So this is a perfect way for her to share more of her research with you all. So Nina, why don't you go ahead and you can start sharing your screen and take it away.
All right, hi. Can everybody see my screen? Excellent. Yeah, so as Rosalind so kindly said, I am a PhD candidate at UC Berkeley, and I study photosynthesis and plants. And so I get to ask bizarre questions that you may never have thought of, like how come plants don't get sunburn? So let's start with thinking about the problem. Here on Earth, we have a major ecological issue, which is that the sun is very bright and light is full of energy, and it can cause harm to organisms like humans in the form of sunburns and even in extreme cases, skin cancer. But plants, they can hang out in the sun all day long and they don't get any of these harmful effects. So why is it that plants are able to do this and we are not? Well, you might be thinking, that's obvious because plants, they can photosynthesize. They are very well known for being able to harvest the energy from light to make their food. And so they must not be able to get sunburns because they're using all of that energy to make sugar and things like that. But humans actually can also photosynthesize. We use the energy from sunlight to make vitamin D, for example. But we can't use all of the energy from light to make our food the way that plants can. So we have to perform photoprotection, which is the mechanism that organisms use to dissipate damaging energy from light in order to prevent things like sunburns and skin cancer. And in humans, there's a photoprotective pigment that you might have heard of called melanin, and there are varying amounts in different people. But plants actually can't use all of the energy that they get from the sun either, because there's just a lot of it. And so they also have to have their own mechanisms for performing photoprotection. And that is what I'm going to talk to you about today. So to give you a little bit of an overview, I'm first going to tell you about how plants actually harvest light for photosynthesis, because it's more complicated than you might think. And then I'm going to introduce you to the light harvesting complex, the molecular machines that both collect and protect plants from sunlight. So it's a bit of a spoiler. I'm going to tell you about the cost of photoprotection in plants and how it might actually be wasting a lot of energy that we could be using to grow food. And then finally, if there's time, I'm going to introduce you to some of my own research in studying photosynthesis and photoprotection, and hopefully using that to improve the efficiency with which we grow our food. So let's start with photosynthesis. Most of you have probably seen this equation in high school or middle school, where carbon dioxide and water take the energy from the sun inside plant cells and use that to produce sugar and oxygen as a byproduct. But I think it's actually more useful in this context to think of photosynthesis as an assembly line, where not only do they need plenty of carbon dioxide and water to make sugar, but also all of the cellular machinery inside of the plant needs to be available in order to use as much of that energy as they can to make sugar and ultimately biomass, which is the stems, roots, shoots, leaves, and tomatoes, and all of those good things that we want to get out of plants as they grow. So let's talk about how this happens and where. Absorption of sunlight occurs in all green tissues in plants, but it mostly takes place in the leaf. And that is because leaf cells contain the highest density of chloroplasts, which are the locations inside of cells where chlorophyll exists. And chlorophyll is that famous pigment that makes leaves green and absorbs all of the energy from sunlight. It does all of the heavy lifting there. But chlorophyll isn't just floating around inside of the chloroplast. It's actually highly regulated. And so the organization of chloroplast is performed by these little molecular machines called light harvesting complexes that arrange the chlorophyll in a very particular orientation. There are millions of these light harvesting complexes inside of the membranes, inside of the chloroplast. And they're composed of three major components. As I said, the chlorophyll, also a protein scaffold, which is shown here in pink in this cartoon, and these structural molecules called carotenoids. And you might have heard of those before because they are the colored molecules that make tomatoes red and carrots orange. And you can see their presence inside of leaves during the fall when chlorophyll starts to get broken down in preparation for winter and leaves start to turn red, orange, and yellow. And so let's talk a little bit more about how this actually works and why I'm calling the light harvesting complex a molecular machine. So I'm calling them that because they're sort of these amazing little transformers that can do both photosynthesis and photoprotection, and they do that by changing shape. So when light is low and limiting, and plants want to use as much of the available sunlight as possible to make sugar, all of the light harvesting complexes are in this on shape. So I have it drawn here as a little tractor to kind of help you understand this like on conformation, which is an actual shape that the chlorophyll takes to direct that energy toward the production of sugar. But when light is high, the light harvesting complexes are actually turned off. And so they're in a different shape or conformation that allows the chlorophyll to direct that energy away from sugar production and also to prevent sunburns. So this is kind of the first answer to our question, how is it that plants prevent themselves from getting sunburned? And that is that the same machine that does light harvesting or photosynthesis also does photoprotection, but it can't do it at the same time. So let's talk about how this actually works. So like I said, chlorophyll is a molecule that absorbs energy from light and directs it towards sugar production. And in, say, limiting light, like on a cloudy day, all of the light harvesting complexes are going to be in this on shape inside of the leaf. And that means all of the energy absorbed by chlorophyll from the sun is going to be directed towards sugar and biomass production. But in excess light, like on a very sunny afternoon, the assembly line for photosynthesis gets very backed up because, like I said, it's not enough for light and carbon dioxide to be in abundance. There are all of these like downstream molecular machines that need to be available to process that energy and make sugar. And when there's too much excess energy that chlorophyll has absorbed, that energy needs somewhere else to go. So can I ask if anybody remembers what the byproduct is of photosynthesis besides sugar? I can't see the chat, so if somebody wants to say out loud or if Rosalind wants to read it.
Oxygen, oxygen.
Oxygen, oxygen, oxygen is the right answer. So there is tons of oxygen floating around inside of cells because it is the primary byproduct of photosynthesis, aside from sugar. And oxygen is actually extremely dangerous to have floating around in a cell because when chlorophyll can't direct its energy towards sugar production in excess light, oxygen is the first thing that's going to want to take up that energy and get what we call excited. So excited oxygen is really dangerous because it's highly reactive and it can tear apart lipid membranes and proteins and even chlorophyll itself inside of the cell. So it's very important that the plant prevent this from happening. And if you think about a plant sunburn, essentially the creation of this excited oxygen and the tearing apart of the sensitive tissues is what it would look like for a plant to get sunburned. And so on one of these sunny afternoons, the light harvesting complexes will turn into this off configuration or off mode, and that enables the chlorophyll to be put into an orientation that dissipates light energy harmlessly as heat. So even though we're saying that plants can't get sunburned, they actually do this by generating heat themselves. And that's preferable to donating all of that energy to oxygen. But I told you also that there was a problem here, and that is that plants are actually too good at photoprotection and too good at turning and keeping their light harvesting complexes off. So even though it's a very quick reaction for plants to detect high light and turn their light harvesting complexes from on to off, turning them back on when shade returns, like a cloud is passing overhead and it would be better to optimize photosynthesis, can actually take minutes to hours. And so you can waste an entire shady afternoon thinking that you're going to get a sunburn and missing out on some valuable photosynthetic time. And this is a huge cost, especially in crop production, because actually 30% of biomass is lost over the course of a plant's life to this failure to transition from photoprotection to photosynthesis when it would be valuable to do so. And that's 30% fewer tomatoes and carrots and soybeans that we could have otherwise gotten out of our crops. And it's also a lot of wasted water and fertilizer that the plant is still taking up over the course of its life, but not turning into those valuable products. So why hasn't evolution solved this problem already? You might be thinking. Well, it turns out evolution really isn't that interested in making plants big. Evolution is actually optimizing for things like reproductive success and survival, and generating a lot of biomass is just something that we want to extract from our crops when we want food and materials and fuel. But there are hundreds of thousands of species of land plants, and beyond that, millions of species of photosynthetic bacteria and algae, and we have not examined all of the photoprotective mechanisms in all of these plants that might have had different evolutionary pressures on them. And so my PhD project is actually looking at this enormous natural diversity in wild plants, in algae, and native crop relatives to find light harvesting complexes that might have evolved to be much better at this transition between photoprotection and photosynthesis in order to make crop production more efficient through genetic engineering and hopefully enable us to use fewer resources to grow our food. So I have some photo credits, and I would like to thank everyone here today who has put this together, including Dee and Roslyn, and also all of the members of the Neogi Lab who have taught me things and have helped me collect plants and work on my project, and the docents at the Botanical Garden who have helped me with a lot of my research. So I will take any questions.
Thanks so much. Wonderful presentation. We got a lot of questions in the chat for you here. You talked a lot about the light harvesting complex changing shape when it senses high light. How does it actually do that? And then we also had another question about why does it take so long for it to change back to the original conformation?
Those are really good questions. So the actual mechanism of how the light harvesting complex changes shape is the major debate in the field. And there are actually many light harvesting complexes in an individual plant, and they all might be doing the same thing a little bit differently. And so one of the mechanisms that's well known is that they actually are able to switch out which carotenoids, those structural molecules that are red and orange, they can actually switch out which of those is creating the structure of the complex, but that's only one of the known mechanisms, or one of the debated mechanisms. And sorry, what was the second question?
No problem. The second question related to that is, why does it take so long for the light harvesting complex to change back into its original conformation?
Right. So if we take the example of the switching of the carotenoids, one theory or one hypothesis that people have is that the mechanism that's well understood is that it's actually very easy to take out the original pigments that are there for light harvesting and put in the photoprotective ones, but doing the reverse reaction requires different genes, and that's actually much more difficult.
Thanks so much, Nina. We had another question. Is this as true in blueberries that have, and I quote, hecka antioxidants?
Great. Okay. Antioxidants. That's amazing. So antioxidants are another way that plants do photoprotection. So people might have, heard of blueberries have like all these great antioxidants, and those are any molecule that can quench excited oxygen, so it can take that excited energy away and return oxygen to its peaceful state, or its ground state. And these antioxidants also occur in other parts of the plant, but they are not as essential to preventing the generation of that excited oxygen. So the photoprotective mechanism I talked about is comes before that. So if that fails, then antioxidants act as kind of the stopgap.
Super interesting. Thanks for sharing a little bit more about that. Another question is, why are you interested in using genetic modification to improve crops rather than kind of those more traditional methods?
Yeah, so I think a lot of people are very scared of genetic modification, but it's actually much faster to create plants that are kind of doing the thing you want them to do by using genetic modification. You can make smaller changes than you can in traditional breeding. So it's faster and it's more specific. And actually it allows you to have a lot more control over the byproducts that might be produced through genetic engineering of a plant. So in traditional breeding, you might be mixing two different breeds of the same species of plant, and you can't control half of what the genome is doing. But in genetic engineering, you can control one gene at a time, which is actually much safer in case like a wild relative that you're interbreeding with has some kind of toxic secondary metabolite or something like that.
Thanks, Nina. Another question that we got is, what's the magnitude of the heat dissipation during photoprotection, and how much does it change the temperature of the plant? Could you explain a little bit more about that?
Yeah, so it's a little bit of a complicated answer. So heat is, if we take a step back, heat is fundamentally the movement of molecules. So when we're talking at the molecular level of how to dissipate energy, it can be released as like light or to another chemical, so you're just transferring that energy, or it can be released through the movement of molecules. And so basically what I'm talking about is that instead of that energy being transferred to another molecule, the chlorophyll is kind of jittering more quickly. But this is measurable. And so I can't tell you about the exact magnitude, but I could tell you that you can tell if a plant is doing photoprotection using like an IR gun. So you can look at like the infrared signature, and you can tell that it is like a measurable difference in temperature, but not enough to be able to feel if you were, say, like touching the plant.
Thanks, Nina. We have a couple of kind of related questions that I found kind of interesting. planting recommendations for many plants are for them to be in full sun, which is about 6 to 8 hours of sun a day. Is there some sort of inconsistency here that, doesn't maybe seem to add up?
Yeah, so I think some things like this has to do with like plants are really complicated and they have a lot of different needs. And so partially it's that natural diversity I was talking about, where photoprotection in all plants is not very well understood. And high light plants, or plants that like prefer full sun, maybe have more photoprotection turned on all the time. And so it's actually more difficult for them to harvest light in general. And so that might be one reason why you have plants that are kind of that prefer full sun.
Thanks, Nina. A kind of related question. How do plants in very sunny environments prevent sunburns without losing too much biomass due to photoprotection? I think that's kind of related to that previous question.
Yeah, so this is something I was trying to get at a little bit in the talk. So how do plants in shade, so sorry, can you repeat the question a bit?
How do plants in very sunny environments prevent sunburns without losing too much biomass due to photoprotection?
Right. So a lot of plants in very sunny environments also do most of their photosynthesis in the early morning or late in the evening, and they're photoprotecting for most of the day. And another reason is, or another way that they prevent that is that they're actually not that concerned about it, or they do most of their growth at different developmental points in their life. So they might grow the most when they're very young and they have most of their photoprotection turned off, and then they don't care about growing anymore once they reach a particular age, and they kind of optimize in different ways.
That makes sense. Thanks, Nina. We had another question. What's happening when the leaves on my indoor plants turn brown? Does this have anything to do with what you're talking about?
It could. So I also am terrible with this, and all of my plants in my house die, which doesn't speak well of what I'm learning in my plant PhD. But there can be a lot of different reasons. So it could be that the balance of water and sun is off. It could be that you have too much or not enough water. But there are plants that prefer growing in the shade. And so if they're getting too much sun, maybe they aren't as good at photoprotection. And in that case, they are getting sunburned.
All right. Thank you so much, Nina. I think we just had one last question here, and then we'll wrap up for the evening. Going kind of back to what you're talking about with genetic modification, when you're doing this modification genetically, are you changing things just within one organism that already exists, or are you adding in something new? Like a gene insertion from like a wild type relative. I think we've got some people more familiar with the subject in the audience.
Sure. So there are a lot of different kind of routes I'm interested in taking. So I'm doing kind of a big survey, but I'm basically interested in taking genes from wild organisms and inserting them into crop plants, so plants that we use to grow food. So, and taking 1 or 2 or 3 genes at a time.
Awesome. Thank you so much, Nina. I just want to say thank you again to Nina Maryn and all of you for being here tonight. So please go ahead. You can check out our website at science@cal.berkeley.edu. We will be sending out a recording later on, probably in early next week, with this recording on YouTube so that you can go back and watch it and share with your friends and families. Be sure to subscribe to our newsletter for all of our upcoming events. And thank you all so much for your great questions this evening. And thanks again to Nina.
Thank you everyone for being here. We hope to see you in person soon. I can't wait to hug you all. Bye everyone.